So hello again. So along the proximal to distal axis in the limb, we have the formation of different structures. But how these different structures are established? So what are the important signals that inform the cells of what they are supposed to do in different parts of the limb, from the proximal to the distal part of the limb? Well, unsurprisingly, or it should be not much surprising to you, there is the formation of a gradient, or actually two opposite gradients, along the proximal to distal axis that resemble very much the gradient that we saw along the anterior to posterior axis in the main body of the animal. In fact, we have a proximal to distal gradient of retinoic acid and a distal to proximal gradient of FGF and Wnt signaling. Now, you should recall that retinoic acid is indeed expressed, produced in the middle somites, and from the middle somites it diffuses inside the developing limb, creating a proximal to distal gradient, while we just mentioned that the apical ectodermal ridge tend to express FGF signaling. This FGF signaling, as well as Wnt signaling, then diffuse as protein from the distal part where the ectodermal ridge is toward the inner part, the proximal part of the limb, creating a distal proximal gradient. Now, this divide the developing limb in different segments, and this, if you want, remind us of the segmentation, or the polarity that we saw forming during the segmentation of the larva of the embryo of Drosophila. So retinoic acid is a signal that represses the apical ectodermal ridge expression of FGF8. So while the ectodermal ridge is near the proximal part of the limb, so when the limb is still very small and is growing, a lot of retinoic acid that is diffusing from the proximal part of the body is repressing the expression of FGF8. However, when the limbs start to grow, and therefore the ectodermal ridge is moved far apart, or more far apart from the proximal part of the limb, it is far away enough so that retinoic acid is not strong enough to repress the expression of FGF8, and the expression of FGF8 also induce the expression on the mesenchyma of inhibitor of retinoic acid. So these inhibitors reinforce even more the distal to proximal gradient of FGF8 and proximal to distal gradient of retinoic acid. What is important to remember, what is also important to remember, is that retinoic acid and FGF8 signal control the expression of very similar genes, very similar transcription factor, like MACE1 and 2 and OX13, but of course in opposite way. In fact, MACE1 and 2 is activated by retinoic acid, while it is repressed by FGF8. And OX13 is activated by FGF8 and repressed by retinoic acid. And therefore, MACE1 and 2 is highly expressed in the proximal part of the limb, so where the stilopod is supposed to be, while OX13 is expressed in the autopod, the distal part of the limb, where we have higher level of FGF8. And in fact, how the cells, or how the different parts of the limb are specified in terms of which part of the limb should become stilopod, which part of the limb should become zygopod, and which part of the limb autopod, is specified by a ratio, by a gradient of retinoic acid, by a ratio of retinoic acid to FGF8. And there are a model that describes this ratio, or change in this ratio along the proximal to distal axis, And in this model, we identified two important points, two important thresholds that when we pass them, we move from one structure of the limb to another. So in the first part of the limb, we have high level of retinoic acid compared to FGF8. So the ratio between retinoic acid to FGF8 is very high, and so therefore the stylopod is produced. However, we reach one point where the retinoic acid is still high, higher than FGF8, but is not so high. So we have the first transition point where we have still high level of retinoic acid, where we move from producing stylopod to producing stylopod. And the second transition point is where we have low level of retinoic acid to FGF8. So the retinoic acid is still there, but not as much, and we have high level of FGF8, so the ratio becomes lower. And this is a transition point where we move from the zygopod to the autopod. So obviously, you notice that depending on which species, you can very easily have a different size stylopod, zygopod, and autopod, just changing the sensitivity ratio of these two transition points. So you can have species, tetrapod species, which have very long stylopod if you move this threshold point to the right. So you can both move this threshold point to the right, so the zygopod will stay the same while the stylopod will become longer, or you can actually move these two threshold points near each other, and therefore producing bigger or longer stylopod and autopod, and shorter or smaller if you want zygopod. So evolution can actually work on this threshold to change the shape of limbs in different species, simply changing the sensitivity ratio of these two threshold points. Now, another layer of expression that controls which bones are formed in a different part of the limb are the expression of OX genes. Now, OX gene expression is controlled by the genes that we mentioned before, that are controlled by retinoic acid and FGF8. Now, remember that when we talk about OX genes, we always need to remember the temporal collinearity. If you remember, temporal collinearity meant that the position of the OX genes in the chromosomes match the expression profile. So the early genes in the chromosome are expressed early during development and more anteriorly, and while the genes that are positioned later in the chromosome are expressed later in the development and more posteriorly. And now this same collinearity is employed here along the proximal to distal axis. And we have that some OX genes are expressed and specify the stylopod, the zygopod, and the autopod. And we have the OX, for example, OX9 and 10 are specifically expressed and specify the stylopod, and you see that they are lower number than the OX genes that are expressed in the zygopod, which is OX11, while OX12 and 13 are specifying the autopod, so they are expressed specifically in the autopod to identify, to specify bones of the autopod. And then in this picture, you can see a nice experiment that demonstrates how the OX genes are important for the morphogenesis of the bones of the stylopod, zygopod, and autopod. On the left, you have a normal-looking bone structure of a mouse forelimb. You have the scapula here. Then we have the stylopod, a normal-looking stylopod, normal-looking zygopod, and normal-looking autopod. On the right-hand side, we have the limb, the forelimb of a mutant, an OX11 mutation. So in this animal, we do not have expression of OX11, or we have very little expression of OX11. And what we find is that the scapula more or less is normal. The stylopod, more or less, is normal, but the zygopod, where OX11 is supposed to be expressed, is very much reduced. And the autopod look almost normal, so there is some effect of removing the expression of OX11 also on the autopod. So this suggests how the OX genes are important in shaping the different elements, the different bonds along the proximal to distal axis of the embryo, of the developing limb. All right. So one region that is particularly important in the progress zone is the zone of polarizing activity. So in this cartoon here, you see these red cells that correspond to the progress zone, which are very active and they are prolifying cell division and so on. One region, the posterior region here, is called the zone of polarizing activity. This region is important to establish an anterior-posterior axis and provide anterior-posterior information. But how do we know that that's the case? Well, one of the first experiments that proved that the zone of polarizing activity is indeed important to establish an anterior-posterior axis is a crafting experiment, or transplant experiment, which is demonstrated here in this picture. So, in this transplant experiment, the zone of polarizing activity, so this region of the progress zone, is transferred from a donor to a host, so from one animal to another. And one of the first experiments was done in chicken. So they transfer this region from one chicken embryo to another chicken embryo. But instead of transferring in the normal position, the transfer or the transplant was put on the opposite side of the progress zone, so in an anterior position instead of the posterior position. And this results in a very bizarre, if you want, limb development. So what we have here is what the result of this transplant is showing this X-ray picture. And what you can see is that the autopod in particular of this animal has been duplicated upside down. So if you look on this particular animal, you see here the stylopod. The stylopod look particularly almost normal or normal. The zygopod look normal, perhaps with some small aberration, but the autopod look completely different. And in fact, what you see here is that the autopod is replicated upside down. So it's like having two hands, one on top of each other in this particular autopod. And this indicates that the signals from the ZPA was actually giving anterior-posterior instruction. Remember that anterior is where the thumb is, and posterior is where the pinky is. So if you have two of these signals, signal center that are competing to each other, what you end up having in this poor embryo is an autopod, which a duplicated autopod upside down. Now, what is the signal that the ZPA is sending to establish an anterior-posterior axis? Well, the signal is SHH. And in fact, misregulation in the limb or during the limb development of SHH results in disruption of the digit production and even having more digit or less digit than normal. And in mouse, there is a particular mutant which is called the hemimelic extratose mutant or mutation that causes a disruption in the expression of SHH from the ZPA. And here you have on the left hand side an X-ray, a picture of a normal looking orthopod of a mouse. Here you have the first digit, the thumb, and this is the pinky. While on the right hand side you have a picture of extra toes in this mouse. And what you can see here is an upside extra orthopod, if you want. This here is a pinky again, so is another pinky. So the toes should be here in the middle. Obviously we don't see them. How the different digits are established? Well, the different digits are established because of exposure or expression of SSH in different parts of the orthopod that is driven by the ZPA. So the identity of the different digits depends on for how long the cell receives SHH or express SHH. Now, in this picture here, you see how the digit 4 and 5 cells are cells that express high level of SHH and are exposed to high level of SHH. And to distinguish digit 5 to digit 4 precursor is that the digit 4 precursor receive or are exposed for less time than the digit 5 precursor cells to SHH. Now, the digit 3 precursor cells express and are exposed to SHH for a shorter period than digit 4 precursor cells, and digit 2 precursor cells do not express at all SHH, but they are exposed to SHH by diffusion from other regions of the limb. So diffusion from the digit 3 region will induce digit 2 formation in those cells. And what about digit 1? Digit 1 developed without any SHH at all. So that the development of digit 1 is independent from the expression or exposure to SHH. And in fact, there are, for example, chicken, birds, mutants which remove the expression of SHH in the zone of polarizing activity. Without that expression, obviously, what's happened is the output produces only a single digit, which is digit 1. And you can see here, if you think about it, you can see here how evolution could very easily intervene to change the number of digits. For example, expanding or reducing the expression of SHH, or for how long is SHH is expressed and how it is diffused. So evolution requires very small changes to change the number of digits. So if there was an evolutionary pressure on the number of digits, this could be changed very easily from 5 to 3 to 1 to 7, who knows what. Okay? So these are all information. These are extra information that I want to give you, just to make you aware how evolution could, in fact, easily modify different aspects of the animal structure, just modifying pattern of expression. It doesn't need to modify gene sequence per se. Okay, so SHH also play an important role connecting the zone of polarizing activity with the apical ectodermal ridge. How this happens? So in the very early limb, you remember we talked about establishing a positive feedback loop between the FGF10 expressed in the mesoderm, so in the proxone, and the FGF8 expressed in the ectoderm. So this FGF10 and FGF8 connect the mesoderm and the ectoderm of the early developing embryo, promoting outgrowing. On a more mature, if you want, limb, where we finally find the zone of polarizing activity, which expresses SHH, we have a connection between the expression of this SHH in the zone of polarizing activity and the expression of FGF8 in the apical ectodermal region. And how this works? So, in the mesoderm, we have expression of BMP. But BMP is, in this case, is an inhibitor of FGF8 expressed in the apical ectodermal region. However, BMP does not work, does not inhibit the expression of FGF8 because SHH induces in the mesoderm expression of gremlin. Gremlin is an inhibitor of BMP. So, because of expression of SHH in the zone of polarizing activity, the mesoderm is not expressing BMP. It is actually expressing gremlin that blocks the activity of BMP. And therefore, because there is not expression of BMP, thanks to the expression of SHH in the zone of polarizing activity, the apical ectodermal region can easily express FGF8, which will be blocked by BMP. So SHH induces gremlin, gremlin blocks BMP, and therefore FGF8 is active, and this promotes the outgrowth of the limb body. On the other hand, FGF8 in the posterior region, FGF8 inhibits the activity of SHH inhibitors. So there are proteins here, genes and proteins that will inhibit the expression of SHH, but in the posterior region, FGF8 inhibits them. However, in the anterior region, the level of FGF8 are different, and this level of FGF8 do not repress the activity of the inhibitors, but actually promote the expression of inhibitors of SHH. So in the anterior region, SHH is not allowed to be expressed, and it is allowed to be expressed only in the posterior region. And this helps forming the SHH gradient that we talked a minute ago, that is important for the definition and specification of the different digits. So what's happening here is an example of how promoter region of different genes can work in a very complicated way. So in this case, we have the FGF8 in one region, probably depending on the level of expression of FGF8 in that region, inhibits the activity of SHH inhibitors, while in another region, the anterior region, the same FGF8, but at other levels, promote the activity of SHH inhibitors. So during the outgrowing, eventually, high level of FGF8 actually trigger a negative feedback loop that blocks the expression of gremlin in the mesenchyma. So at one point, the apical ectodermal ridge starts to be separated or far away from the region of expression of gremlin, and start to produce a lot of FGF8. And if there is a lot of FGF8, high level of FGF8, this induces a repression, if you want, of gremlin 1. It's like, in this case, a negative feedback loop that controls the expression of FGF8, so that the amount of FGF8 can be blocked, if you want, or finally monitored using this negative feedback loop. Because when there is a high level of FGF8, which will block the expression of gremlin. And remember, gremlin was the one that was blocking activity of BMP. And BMP is that gene, that protein, that will block the activity of FGF8. So in this case, when we have a lot of FGF8, this will induce blocking of gremlin. So gremlin is not there, and then BMP is active to block and reduce the expression of FGF8. So what's happened? The repression of gremlin. So when we have a repression of expression of gremlin, and we have also an increased distance between the mesenchyma that is used to express gremlin, but also an increased distance between the R and the ZPA. So the two signaling centers are far apart, so they cannot really help each other anymore or sustain each other anymore. When the gremlin goes down, so because the mesenchyma doesn't receive too much SHH anymore, and it does receive a lot of FGF8, which will repress gremlin, and then therefore allowed BMP expression. So what's happened when we have BMP activation? BMP activation repress FGF8 in the apical ectodermal ridge. Without expression of FGF8 in the apical ectodermal ridge, there is no more signaling that goes and help the zonopolarizing activity to express SHH. So SHH is not expressed over there, and therefore the zonopolarizing activity is terminated. Without zonopolarizing activity, there is no more sustained signals that will allow the expression of FGF signals in the apical ectodermal ridge, and also the apical ectodermal ridge at this point is terminated. So when both the ZPA is shut down and the area and the apical ectodermal ridge is shut down, when both of these are shut down, the embryonic development of the limb is terminated. So at this point the limb is elongated, the digits are formed or are in the process of being forming, but they cannot be anymore sustained and fast outgrowing of the limb. So now, the only axis that we need to talk about, that is left to talk about, is the dorsal ventral axis. In this cartoon here, you see this circle, this ball that represents the different somites. You have anterior position on the top, posterior position above, and here is the limb that is developing. In red, you have the apical ectodermal ridge that separate the dorsal portion of the limb from the ventral ridge. Now, how the dorsal ventral axis is determined depends on the surrounding ectoderm. So it is the ectoderm, the surrounding limb, that is defining the ventral and dorsal portion of the limb. Now how this is happening? Well, the dorsal ectoderm, the dorsal ectoderm express Wnt7. So we know that the ectoderm express Wnt signaling. And in this case, the dorsal part of the ectoderm of the limb expressed Wnt7. Wnt7 is only expressed in the dorsal part of the limb, but not in the ventral part. Wnt7 induced expression in the below mesenchyma, in the dorsal mesenchyma of limb 1, or LMX1b, or limb 1 for simplicity. So limb 1 is expressed in the dorsal mesenchyma, but is not expressed in the ventral mesenchyma. And in fact, the ventral mesenchyma express BMP, express a lot of BMP. And if you remember, we just mentioned that when we have expression of BMP, or we have a lot of expression of BMP, we are shutting down the apical ectodermal ridge and the zonopolarized activity. Okay, so in the ventral region we have expression of BMP in the mesenchyma. This expression of the mesodermal BMP inducing the ectoderm expression of engrade 1. So the ventral ectoderm express engrade 1, while the dorsal ectoderm express Wnt7. So now we have a formation of two distinct parts between dorsal and ventral. The dorsal ectoderm expressing Wnt7, the ventral ectoderm expressing engrade 1, and then a gradient between dorsal and ventral of limb 1 and BMP. BMP is responsible for, so it is connected with the axis, because BMP is responsible for shutting down the apical ectodermal ridge. It indirectly is therefore responsible for shutting down the zone of polarizing activity. It also inhibits Wnt7, so it is responsible for the formation of the dorsal-ventral axis in the limb. So BMP is somehow connected with all three axes: the dorsal-ventral directly, the proximal distal indirectly, as well as the anterior-posterior indirectly. Okay, so I hope that after revising this lesson, you will be able to describe how the limb bag is established, how the OX genes and different other genes are controlling the different structures that are forming the limb, and importantly, how retinoic acid, FGF signaling, Wnt signaling are connected, and SH8 signaling as well, are connected to each other to create the different gradients, the proximal to distal gradient, the anterior-posterior gradient, and well, dorsal to ventral gradient in the limb during the very early development.